Hello, Melissa. My name is Zohra. Today, Mikhail and I are going to talk about the impact of temperature on yeast fermentation and identifying the optimal temperature for CO2 production. Next, please. I'm going to talk about introduction and I will continue with method. And Mikhail is going to talk about results and conclusion. Next, please. Yeast are single-cell fungi. Like other eukaryotes, they use O2 to produce ATP as their source of energy. They produce ATP through oxidative phosphorylation or through fermentation. Fermentation is a process that occurs in cytoplasm, and 2-ATP is generated. In addition to ATP, CO2 and ethanol are produced by fermentation. Yeasts are temperature sensitive, and if the temperature is not optimal, the amount of CO2 and ethanol may be affected. Because fermentation is enzyme driven, and enzymes are sensitive to temperature. Increasing temperature above optimal temperature affects enzyme sterility shape and may denature their shape. For this experiment, we considered how temperature affects CO2 production and what the optimal temperature is. Next, please. Um, the method we used for this experiment was inverted test tube. And the yeast we selected was Baker's yeast. We carried out the experiment at 25, 35, 50, and 65 degrees Celsius. First, 7.5 milliliter of 1% yeast was added to 7.5 milliliter of 1% glucose. And for each temperature, we did 5 replications with 5 test tubes and 5 conical tubes. The amount of CO2 was measured in milliliter at 5 minutes interval for 30 minutes. And Mikhail now will continue the rest.
Okay. So we try to maintain consistent volume of yeast and glucose solution in all test tubes. We ensured water baths were accurately set to specific temperature. We tried to conduct measurements at standardized time intervals to eliminate variability, and we implemented multiple replicates to validate the reliability of results. So for results, we calculated for every 5 minutes intervals the mean production of carbon dioxide for each temperature. The highest at 50 degrees Celsius, which is 5.9 milliliter, and the lowest is at 25 degrees Celsius, which is also a room temperature uh, after 30 minutes. So for the as we go in the table showing the concentration of carbon dioxide at 25 degrees Celsius, 35 degrees Celsius, 50 degrees Celsius, and 65 degrees Celsius. The concentration of carbon dioxide at 35 degree and 65 degree is almost similar. For 4.12 at 35 degree and 4.50 is 65 degrees Celsius. But in the case of 50 degrees Celsius, the production of carbon dioxide is high in comparison with both of them, which is 7.70. In this table, we also measured the 95% confidence from our data. Here we have the graphical presentation for results. This is a graph for 95% confidence respect to temperature. We can see the 35-degree bar and 65-degree bar are overlapping each other. So it indicates no significant difference in carbon dioxide production. But as we can see, at 50 degrees Celsius, there is a drastic increase in the production of carbon dioxide. So border 95% confidence intervals at 65 degrees Celsius, indicating higher variability in our data points. So for conclusions, we can say yeast are eukaryotic cell. As we know, eukaryotic cell use enzyme to perform fermentation. So enzymes are sensitive to temperatures, and to have maximum amount of carbon dioxide, they need to be operated at their optimal temperature. That's for Baker East is 50 degrees Celsius. This is our reference we used for our research, and thank you all.